Hello guys and welcome back to another video from my YouTube channel, that is Vipin Sharma Biology Tutorial. And today we are talking about the structure of cell membrane or plasma membrane, which forms a very important question in all pre-medical examinations. So firstly we have to understand what is the plasma membrane or cell membrane. If we are considering a cell, for example this is a cell, then the outer covering of this particular cell is called as the plasma membrane. This is the nucleus, centrally located nucleus. Okay, so this is a cell and this particular region is known as the cell membrane or plasma membrane, and this is the nucleus. This region, which has different kind of granules or organelles, is known as cytoplasm. So this particular region is known as cytoplasm. So after that we have to emphasize on the structure as well as functions of plasma membrane. So firstly we are going to look at some important diagrams of new NCERT book. After that an diagram of old NCERT book, and after that an example from graduation book. और उसके बाद हम देखेंगे कुछ notes जिसमें हैं all the important points for plasma membrane which may be asked in examination this year. So we are going to emphasize on each and everything about plasma membrane because we can't take a risk. Okay, so let's start. This is the structure of cell membrane which is given in new NCERT book. Okay, so what kind of structure it is? It is the fluid mosaic model of plasma membrane. This particular model of the structure of a plasma membrane is given by two scientists, that is Singer and Nicholson, in 1972. This forms a really very important question, that who proposed the fluid mosaic model of plasma membrane. We are going to see in next slides, that is in the notes that are made by me, that this particular model is given by Singer and Nicholson in 1972. So what is the fluid mosaic model? These particular circles, these are the polar heads of lipid layer. This is a lipid layer and this is a second lipid layer. These are the polar heads and these blue circles are also the polar heads. After that there are some tails. So the head is polar, the tail is non-polar, and the head is hydrophilic because this extracellular space is having water, and that particular water is interacting with the outer surface. So the outer surface would be hydrophilic, that is water loving. So the outer surface is water loving. Okay, and the surface that is present inside, that is this particular region, that we are calling the tail. These are the hydrophobic tail because these are present in opposite direction to the extra cellular substances, that is water. So they are repelling water, therefore these are hydrophobic in nature. So the tails are hydrophobic in nature. After that there are different kind of proteins which are either peripheral, that are present in the periphery. For example, this is a particular protein which is present on the periphery, that is only on this membrane, above this membrane. And there are some sort of proteins which are present in the integrated manner. It means these are present embedded between the two layers of bilayers of lipid. This is one lipid layer, this is another lipid layer, and these are some proteins. These are some proteins which are known as the integral proteins. The integral protein, they are known as integral protein because these are present in an integrated manner, or they are embedded in the two different phospholipid bilayers. Phospholipid bilayer because these are lipid in nature, lipid bilayer, and it contains phosphate residues. So this is also known as phospholipid bilayer. Okay, that is written here, that is lipid bilayer. Two layers of lipid, one is this layer and one is this layer. Okay, so after that there are different kind of proteins which we have talked about, that are peripheral proteins or the integrated proteins. Except these, there are some other kind of proteins which are present here. For example, this is an alpha helix. We know that if we are talking about the sequence of amino acid, Amino acid are the monomers of protein. So protein is formed up of different kind of amino acids. So the amino acids, if are present in a single line, then that is the primary structure of protein. And in the secondary structure of protein, they form two different conformation. One is in the form of sheets, that is known as beta pleated sheet, and another one in the form of a coil, and that particular coil is known as the alpha helix in the presence of in the form of a helix. So here we can look that this particular structure is present as a helix. This particular structure is present as a helix in a coiled manner, as we can see. So this is a alpha helix protein. After that, there are some protein residues. For example, this. For example, this. These are also different kind of proteins. They may be alpha helix, beta coiled, beta pleated, and different kind of proteins, turns, loops, helixes. So there are different kind of or different conformations of protein which may be present in the plasma membrane. After that, there are also some sugars. Sugars. This is sugar. This is sugar. In this particular manner, branch sugars. For example, this. So all these sugars are also present in plasma membrane. There is a membrane stabilizer, a particular chemical that stabilizes membrane, and that particular membrane stabilizer is known as cholesterol. That particular thing is also present in the membrane. This particular granules are the cholesterol granules. After that, the combination of sugar and protein. Sugar means glyco. The word glyco is used for sugars, and proteins are simply proteins. So in this particular case, the protein is integrated with sugar or linking with sugar. So it is known as a glycoprotein. And in some cases, this particular sugar is integrated with or binding with lipid molecules. In this particular case, this is known as glycolipid, since glyco means sugar and lipid means lipid itself. So these are the different combinations of sugar with proteins and lipids, that is glycoprotein and glycolipid. The next diagram is from the NCERT book, the old NCERT. So this diagram is very rare, so we have to emphasize on that also. This particular protein is protein channel or transport protein. Okay, so since the plasma membrane also helps us in the transport of a particular particle from this direction to this direction, that is across membrane. So there are some chemicals which are transported via the help of some carrier proteins which are known as permeases. These are known as permeases, which are totally embedded in between the two lipid bilayers so that they can transfer a particular substance from one direction to the other direction. So this is known as the carrier protein. Okay, or the transport protein. The different proteins that are peripheral in nature are also globular protein. So this question has arisen many times, but most of us think that this question is out of NCERT, but actually this is from the previous NCERT book, that is old NCERT edition. So these are some globular proteins. You have to note that. After that, we have already talked about glycoprotein, that in this particular case, the carbohydrate molecules or the sugar molecules are integrated or are linked with this particular thing, that is protein. So glycoprotein. There is extracellular fluid. Okay, now carbohydrate we have done. These are hydrophilic head because these are facing the water. This is phospholipid bilayer having two layers. One is this one and another one is this one. Okay, and a particular molecule in this particular bilayer, there is a polar head and non-polar tails. So this particular molecules is known as a phospholipid molecules. After that, we have talked about the alpha helical protein molecule. We have talked about hydrophobic tails. There below this particular cell membrane, inside the cell, if the upper side is the extracellular fluid, then the inside, if this is the extracellular fluid, then this particular side will be having the cell organelles, that is cytoplasm. Okay, so this particular side will be having some filaments, some cytoskeleton, as well as different kind of organelles. This particular protein is the integral protein. or transmembrane protein. Why it is known as transmembrane protein? Because this is crossing the particular membrane from both the sides. This is appearing from this side also, and this is appearing from this side also. Therefore it is known as transmembrane protein as well as tunnel proteins. And why these are known as tunnel proteins? Because this particular molecule of protein is responsible for the transfer of different kind of atoms from one position to the other, as in this case. So they are known as the carrier molecules or the tunnel proteins. So this is the diagram from the old NCERT edition. After that, for simplifying this, I have taken another diagram from another book of graduation level, so that you can easily understand the concept behind the fluid mosaic model of plasma membrane. And after that, when you will reading some sort of notes, that will be very beneficial for you, that all the things must be on your tips. Okay, so let's study this diagram. Okay, so this particular diagram, this is the phospholipid. We have talked about a single molecule of phospholipid having a polar head and two non-polar tails. So this is a lipid layer, this is another layer, so both of them combine to form a phospholipid bilayer. This is a glycolipid, as the sugar molecules is interacting with the lipid molecules. So this is glycolipid. This is transmembrane protein or integral protein, as it is integrated in this membrane, and transmembrane because this is crossing the particular membrane from both the sides. These are some sort of membrane stabilizer which are present in lipid bilayer, that are known as cholesterols, that are most common substances in the you know lipid bilayer. After that, glycoprotein. This is the protein, extrinsic protein, because it is present in the external side, outer side of this particular membrane, and this is a sugar molecule, so it is known as glycoprotein. Okay, so after that, here comes the extracellular space. It means that on this particular side there is cytoplasm, therefore it must be containing cytoskeleton, different kind of fibers, etc. Cellular organelles on this particular direction, that is downer side, sorry, on the this side. This is a particular carbohydrate molecule which when linked with the lipid forms glycolipid, and when it links with the protein it forms glycoprotein. Okay, so this is the structure of phospholipid bilayer. Now some notes from NCERT books, we have to emphasize on that. So let's study some important points. The detailed study of this particular plasma membrane has taken place after the discovery of electron microscope, since to study plasma membrane is not an easy task. To you know its diameter and to know the length and width of different kind of phospholipid molecules, it's very difficult because they are very minute in size. So these all tasks can be possible after the discovery of electron microscope, and that particular discovery was held in 1950s. Okay, so this is very important. After that, the study of that particular phospholipid bilayer or the plasma membrane has taken place for the very first time in RBC, and the particular membrane that is present in RBC is known as the Donan's membrane. So the firstly it is discovered in Donan's membrane, because the first and the most important test for the discoveries of plasma membrane were taken place on RBC, that are red blood cells or erythrocytes. So this is the structure of a particular phospholipid bilayer. Here this is polar head which is facing the extracellular fluid. This is hydrophobic tail. This forms a particular lipid layer, and this is also another layer, so it forms a lipid bilayer, phospholipid bilayer, because the phosphate molecules are also present. These are non-polar and saturated. These particular tails are non-polar as well as saturated. What do you mean by saturated hydrocarbons? That there is no double bond present in these particular tails. This is a particular phospholipid molecule in which the tails are saturated. If it is non-saturated, that is the presence of double bond is there, then it forms a kink. Okay, the another tail would be like this. So this particular diversion in the shape of you know that particular tail is known as kink. This is very important. So most of the particular phospholipid present in the phospholipid bilayer are saturated, that is having single bonds only. So these are represented like this in a straight manner. After that, the lipid component has phosphoglycerides. There are some sort of glycerides, means some kind of sugars which are embedded in lipid, that are glycolipids, and phosphate, because we are talking about phospholipids. So these are some constituents of the plasma membrane. After that, the plasma membrane has also contain protein and carbohydrates. So how can we justify this statement? We have talked about the proteins, that is integral protein and peripheral proteins, and we have talked about carbohydrates that are getting linked. The carbohydrate simply means polysaccharides or sugar, which are getting linked with lipid and protein to form glycolipids and glycoproteins respectively. And since we have done all these discoveries on RBC, so if we'll talk about RBC, then the protein content, since the plasma membrane is formed up of lipid and protein, so the protein content in the RBC is 52% and the lipid content is 40%. So the protein is 52% and the lipid is 40% in the phospholipid bilayer integrated with protein, or we can simply say it is the plasma membrane of RBC. So this is about the plasma membrane, that what kind of proteins does it have. So the next slide is this. In this particular section we are going to talk about different kind of proteins that are present in plasma membrane. Okay, so on the basis of ease of extraction, यानी कि किस प्रोटीन को कितनी आसानी से निकाला जा सकता है किसी प्लाज्मा मेम्ब्रेन से, उसके बेसिस पे प्रोटीन्स को डिवाइड किया गया है पेरिफेरल प्रोटीन. पेरिफेरल प्रोटीन्स आर दोज प्रोटीन विच आर प्रेजेंट on the surface of phospholipid. This is a surface, and this particular protein is the peripheral protein, as this is present on the periphery of you know the phospholipid layer. And some are integral proteins, as they are embedded in the phospholipid bilayer. So this is very difficult task to separate the integral proteins from the phospholipid bilayer, but this is very easy to separate that particular peripheral protein from the phospholipid bilayer. So these are depending on the ease of extraction, so they are termed as peripheral and integral. So the fluid mosaic model is given by Singer and Nicholson in 1972. We have discussed that the fluid, it is known as fluid model because the fluid means quasi-fluid lipid. The quasi-fluid lipid is present in this particular membrane, which is responsible for the cell division, because it is the flexibility is required for the cell division as well as cell growth. It is also important for secretion, intercellular junction, it forms the junction between two cells, and for endocytosis. Endocytosis is the engulfment of a particular material. For example, this is a material and this is a cell. This cell is going to engulf this particular material. We have studied about phagocytosis. So this is endocytosis, in which such kind of vesicles are formed, and that vesicles got separated and the thing is engulfed and enter into the particular cell. This is known as the endocytosis. So for all these purposes the flexibility is needed in a particular membrane, and that particular flexibility is provided by the lipid layer, because the lipid layer is quasi fluid in nature, and fluids are a little bit flexible in nature. So this is very important. After that, their mosaic means protein. The proteins are spreaded in the plasma membrane like mosaic. These are present in different kind of positions, and these proteins moved within the layers of membrane, and this particular thing is called fluidity. The one protein can go from this place to this place and from this place to this place. And how can it move? It can move by the virtue of quasi-fluid lipid, because the fluidity of a particular lipid molecules allow the protein to change its position. So this is the fluidity of a particular membrane. After that, the plasma membrane is semi-permeable. Semi-permeable means that it allowed some kind of specific particles to pass through them. So now let us study the function of plasma membranes, that how it allow different kind of transport if this is a particular plasma membrane, so there is some sort of passive transport. Passive transport means the transport across the concentration gradient. So neutral molecules got transported via this membrane from a region of high concentration to a region of low concentration automatically via the concentration gradient. This particular process is known as the diffusion. And the moment of water, if the substrate is water, then that particular process or the passive transport is known as osmosis in case of water. If we will talk about the facilitated diffusion, that is another kind of diffusion. So in case of facilitated diffusion, this is the plasma membrane, and there must be a carrier protein, and that carrier protein will help to transport some kind of particles from one side to the another side of this plasma membrane. This is the plasma membrane. So this particular transport takes place via the help of some carrier proteins which are known as permease, and some polar molecules got transported via this particular mechanism. After that, if we talk about active transport, in active transport the energy should be given to the particular system to transport a particular thing. For example, sodium potassium ATPase. This is the structure of sodium potassium ATPase, which is known as NAK pump. Pump is an important word which is used for the utilization of ATP. It means pump simply means that the ATP is getting involved in this particular process. So since ATP is used, then this is known as the active transport. And in this active transport, the transport of a particular material takes place from low concentration to high concentration, that is against the concentration gradient. So this is very important in this particular type of transport which is known as active transport. So guys, this is all about the structure, functions and different kind of points and all the things about the fluid mosaic model of plasma membrane and the different structures that are given in new NCERT book, old NCERT book and some sort of refresher books for plasma membrane. So I hope that you like this video. So thank you so much guys for watching this video, and if you are new, then